Well, hello, welcome back to another segment for our solutions unit. I guess we're calling this part 7. This is actually kind of a follow-up to what I was describing with the egg. So we saw the egg shrink or expand based on whether water wanted to move into the egg through the membrane or whether the water wanted to come out of the egg through the membrane. And that had everything to do with the solution that egg was sitting in. So there is a measure that solutions have that kind of describes whether water will move out of through a membrane or in through a membrane. And typically this is used in the biology field. So I'm actually going to use a biology example, and I'm told by your biology teachers that you will see this again next year. And so I think it'll be nice because you'll have some background, and you might say next year, like, oh yeah, I remember this from when I was learning about tonicity at home. At least I kind of hope you'll remember. I don't know. Teachers always think you'll remember the great things that they're trying to teach you. It sounds kind of scary because you've probably not heard the word tonicity before, but you don't even need really a definition for that. So let me just show you a diagram here that describes everything. And again, think back to the egg demonstration, and I think it'll make sense. So if we imagine that instead of an egg, we have a cell. Now, do you know, I think you know, that all living things are made of cells, and those cells are a lot of water, but then there's a lot of other things in there too. So cells have membranes that are also semi-permeable. That means water can move usually in and out of them pretty easily, but other larger molecules can't. And sometimes it just depends on what the molecule is. But if you put a cell into a solution, one of three things can happen. The best situation is that the concentration of the stuff in your solution matches the concentration of the stuff in the cell. And then everything's good. We call that equilibrium, and we refer to that, that solution as isotonic. Iso means the same, okay? The same concentration. So in this first figure here that I'm circling with my mouse, we don't need to worry about what the stuff is. We'll just call it a solute. So if there's 20% solute in the cell and there's also 20% solute in the solution around it, then there's no reason that water wants to move in or out because we already have everything in equilibrium. So that's awesome. But what if the solution has a higher solute concentration? Okay, so you see here these little green things represent what that solute is. And there's a whole lot more of them there. So it's very concentrated out here, but very dilute in here. Okay, so what we need to do is we need to try to dilute some of that very concentrated solution. So we call it hyper. Hyper meaning too much, you know, like a hyper person, too much energy, right? Hypertonic means it's got too much solute in it. So we're going to try to dilute it a little bit by getting some water into that solution. Well, where's the water going to come from? It can come from inside the cell, because that also, remember, makes the cells more concentrated, right? So instead of maybe a 20% solute, it's going to rise up to something higher than that. And the 40% concentration that we have in our hypertonic solution is going to come down a little bit, and water will stop moving out of the cell once we reach the same concentration. Now, what is that concentration going to be? Well, it depends on how much solution you have and how big your cells are and how many cells you have. Again, that's too much detail to worry about here. But this is something that doctors and nurses, some of you may want to work in the medical field someday, you have to understand that, because, I mean, there's a constant sort of a balancing act happening within, you know, living things. So we call that hypertonic. Too much is hyper. Too much solute in our solution. But what if the solution is too dilute? Then we call it hypo. And I don't really like that we have hyper and hypo because they're so similar sounding, it's easy to get them mixed up. But don't blame me, I didn't name it. I'm just telling you what it's called. So if the solution that the cell is sitting in has a lower solute concentration, that means it's very dilute, then what we have to do is try to dilute the cells itself. So we move some water into the cell, diluting the solute in there, again, hopefully attempting to reach some equilibrium. And so trying to make everything become an isotonic situation would be ideal. Okay. Now again, a lot of detail here that I'm not going over, but all I want you to do is to be able to describe when a solution is hyper or hypo or isotonic. And then from that predict, okay, does that mean that water will move out of the cell and into the solution, or out of the solution and into the cell? Okay, that's your main skill. And again, this explains everything we saw with the egg, right? It was all about the concentration of the sugar in the case of why the egg shrunk first, and then the concentration of I guess everything else when we saw it expand again. So I'll let you kind of stop it here, get those notes down. Really, that little diagram, don't try to draw it perfectly or whatever. Have yourself a diagram, a sketch that kind of makes sense to you, and you may always have this in front of you or have a tab with it as you're working on the little Google form worksheet, because this is too much right now to necessarily commit to memory. As long as you can kind of see the connection between the ones I'm showing you for practice and these here, you will be golden. Okay, so I'm going to let you kind of stop it here and then carry on for your practice. And as always, let me know if you have questions about anything. Thanks for watching.